Hello everyone. I'm very glad to welcome you all to the second lecture of this online course, Genomics in Immunohematology. So in today's lecture, we'll be learning a lot about the genetics of human blood groups. So starting with our topic, like we had discussed in our introductory session, RBC antigens or the blood group antigens are expressed on the surface of human RBCs. And like we see in the figure here, there are various types of RBC antigens like the ABO, Bombay, RH, Diago, Duffy, which are expressed in a transmembrane fashion in the human RBC. So the main aim of this lecture is going to be understanding how are these blood group antigens formed and what is the role of genetics behind the formation of these RBC antigens. So let us move ahead, and to understand this with more clarity, we are going to take the example of the very well-known A and B antigen scenario. So talking about how are these antigens formed, initially, we have the RBC with a precursor antigen, which is the H antigen. So, to this particular RBC, there are two possibilities. There are two possibilities for the addition of two different donor substrates with the help of two different enzymes. So, the first possibility is that to this particular RBC, a donor substrate called as UDP-N acetyl galactosamine can be added with the help of the enzyme called as A-glycosyl transferase or the GTA enzyme. So this will in turn lead to the expression of the A antigen on the surface of this particular RBC. And the second possibility is that a different donor substrate, which is UDP galactose, can be added to this precursor molecule with the help of an enzyme called as B-glycosyl transferase or the GTB enzyme, which could lead to the expression of B antigen on the surface of this particular RBC. So with this very quick illustration, one thing becomes very much clear. So the difference in the expression of A and B antigen depends upon which donor substrate is added to the precursor molecule with the help of what enzyme. So to know more about what decides the presence of a particular enzyme or the addition of a particular donor substrate, we need to know the genetic basis of what happens on a molecular level. So here we are going to discuss what could be the molecular mechanism behind this difference or how does it get differentiated. For this, we are quickly going to discuss about one of the earliest studies which was done in the early 19th century by Yamamoto and his colleagues. So what basically was done in this study was that multiple cell lines were established from different types of donors who had different ABO phenotypes, like the A phenotype, O phenotype, AB phenotype, and B phenotype. And from all these cell lines, mRNA was extracted, and the main aim of the study was to detect the amino acid sequence for each of these different types of ABO phenotypes, from which they wanted to understand and they also wanted to compare how similar or different their coding sequence looked like. So this very quick and the earliest the earliest study gave some interesting results. The first thing being that the expression or the production of the enzymes, the GTA and the GTB enzymes, was primarily regulated by a gene called as the ABO gene, which was present in chromosome 9. This particular ABO gene comprises 7 exonic regions, and a single person, a particular individual, can have a combination of 3 major alleles. The first one being the A allele, which is responsible for the production of glycosyl A transferase or the GTA enzyme, or the B allele, which is responsible for the production of glycosyl B transferase, which is otherwise called as the GTB enzyme, and the last one being the O allele, which lacks both of this enzymatic activity. So now that we know there are 3 different alleles which plays a major role in deciding what enzyme is produced by a particular individual, the next step is to know how are these alleles categorized or how are we going to identify these 3 different alleles. For that, we are going to put all the findings together in a bigger picture. So first is initially we have chromosome 9, in which we have a particular gene called as the ABO gene, which is responsible for the production of these enzymes. And we all are aware that human beings carries diploid chromosomes. That is, we have two sets of chromosomes which we obtain from our or both the parents. And these are called as allele 1 and allele 2. And talking about the ABO scenario, a particular individual can have these different types of combinations. Like, for example, a person can carry two copies of A allele, or they can carry one copy of A allele and another copy of B allele, or it can be a combination of multiple other sets also, like a O and A, or it can be two copies of B allele. It can be a combination of a O allele and a B allele. Now, going into this with a little bit detail, taking the very first example, there are two copies of A allele, and we know that A allele is responsible for the production of GTA enzyme. So, in the first case, the enzyme which is produced is GTA. Whereas, when we come to the second example, there is one copy of A allele and another copy of B allele. So, there will be a production of both the enzymes GTA and GTB. And coming to the third example, there is one copy of A allele and another copy of O allele. And we know that O allele lacks both the enzymatic activity, so there will be no enzyme produced. So the person will have only the GTA enzyme because there is one copy of the A allele. So in a similar fashion, depending upon what is the combination of alleles present in an individual, it decides what kind of enzyme is being produced by the particular individual. And now that we know what kind of enzyme is present, we will be able to know what kind of donor substrate is going to get added to our precursor RBC. So starting with the first example again, since we have the GTA enzyme, the antigen which is going to get expressed is the A antigen. And in the second example, since there are both the enzymes, GTA and GTB, the antigens which are going to get expressed is both the A and B antigen. And in the third case, it is again going to be the A antigen. So in a similar fashion, depending upon what enzyme is present, in each of this scenario, we will be able to predict what could be the antigen which is getting expressed on the surface of that particular individual's RBC membrane. Having said that, now we are able to have a very clear picture on how the genetic background decides what kind of antigen is going to be expressed on the surface of a particular individual's RBC. And here we are going to note one more important concept. Like we see here, if a person carries two copies of the same allele, like which is highlighted in these yellow boxes, this particular state is called as a homozygous state. Like for example, the person carries two copies of A allele, or two copies of B allele, or two copies of O allele. So this is called as a homozygous state. Whereas in rest of the cases where it is highlighted by the blue box, there are a combination of alleles, combination of different alleles, and this state is called as the heterozygous state. So overall, there is a clear picture that the genetic basis decides what allele is present in a particular individual, and based on what allele is present, the enzyme is decided, and the enzyme which is secreted decides which donor substrate gets added, and this finally decides which antigen is expressed on the surface of the particular individual's RBC surface. So now getting into a little bit detail of how this initial level, the genetic level, looks like or how it differs, let's take a very quick illustration. So here again we have the ABO gene. This is going to be the coding sequence of the ABO gene. And for a better clarity, let us consider some positions like position 261, 297, 526, 657. These are all the positions which are very much important for distinguishing the alleles, distinguishing the A allele, B allele, and the O allele. So let us see how are we going to distinguish the different types of classical alleles. So initially we have the A allele. So at position 261, the A allele carries the G nucleotide, and at position 297, it carries a nucleotide. Similarly, at position 526, it carries the C nucleotide, and at position 657, it again carries a C nucleotide, and so on. And now when we have a look at the B allele, at position 261, again it carries the G nucleotide. Whereas when we look at position 297, it carries the G nucleotide, whereas the A allele carried A nucleotide. So this is how the nucleotide differs between each of the alleles. And similarly, when we take a look at the O allele, when we look at position 261, both the A and B alleles carried the G nucleotide, whereas there is a deletion in this particular position at the OLE. So this is how nucleotide changes. So it can be a single nucleotide variation, like they're changing from A to G or C to G, or it can be a deletion, or it can be an insertion, which distinguishes the A allele from other two alleles, that is the B and O allele. So all of this example is just for one blood group system, which is the ABO blood group system, and the expression of just two antigens, the A and B antigen. And if we quickly recall what we learned in our introductory session, there are more than 300 blood group antigens which may be expressed on the surface of a human RBC. And for each of this antigen, there is a molecular mechanism behind it, and there is a genetic basis which regulates and controls the expression of these antigens. So having said that, if we put all of this together into a bigger picture, for more than a decade, during the early 19th century, there were a lot of studies which explored in uncovering the genetic background of numerous human blood group systems. And currently, we have about a total of 51 unique genes, which we call as the human blood group genes. And these genes can carry a range of genetic variations which could be categorized as single nucleotide variations, it can be deletions or insertions, and all of this lead to a variable expression of antigen on the surface, which will in turn decide the variation in the blood phenotype of an individual. So putting all the pieces of this puzzle together, we have a very clean bigger picture. That is, there are a total of 51 blood group genes, and there are, in all of these 51 genes, there are a total of 1,873 genetic variations. And these variations, like mentioned earlier, can be any type of variation, like single nucleotide variations, insertions, or deletions. All of this together are responsible for the expression of a total of 349 blood group antigens. And based on which gene regulates and which gene carries these genetic variations, they are systematically categorized into something called as blood group systems. And today we have a total of 44 blood group systems, which corresponds to a total of about 634 blood phenotypes. So this is the complete bigger picture of the blood group antigens which are expressed on the human RBCs. Now that we have discussed about this in a greater detail, we also need to know the importance of resources and repositories which maintains this information so that they can be studied in a greater detail. So one of the pioneering bodies which systematically maintains the information of these genetic variations and their associated phenotypes is ISBT, the International Society of Blood Transfusion. So they collect the literature and case reports which talks about newer and novel blood group antigens, and they systematically maintain a data compendium. And whatever information goes into this ISBT resource can be classified into four major categories. That is systems, collections, 700 series, and 901 series. So systems comprises of all those antigens for which a very clear genetic background has been studied, explored, and validated. The next level is the collections, where it comprises of antigens that are serologically very much distinguishable, but their genetic background is yet to be worked upon. And finally, we have the 700 series and the 901 series, which comprises of antigens which are of lower incidence, that is less than 1% in the population. All of these low incidence antigens are compiled together in the 700 series, and all the high incidence antigens, it is greater than 90% in a population, they are put together in the 901 series. So this is how every new findings related to this blood group genetic variations are categorized and systematically maintained by the ISBT. Other than ISBT, also there are many other resources like Erythrogene, the Human Rhesus Base, the Blood Antigen Atlas. These are some of the other sites which comprehensively maintains the different findings which are being published on a day-to-day scale related to the blood group antigens and their corresponding phenotypes. Quickly taking an example of how the data looks like in these ISBT resource, we have here, there are three different antigens: the A, B, presence of both A and B, or absence of both A and B, which we call as the O phenotype. And like we see in the example here, we could clearly see that a particular genetic variation which is getting associated with a particular phenotype. Like for example, there is A1 phenotype, and we could see that there is a SNV which is associated with a particular phenotype, 467C2T. This particular SNV is associated with the A1 phenotype. Similarly, we have the A2 phenotype, A weak phenotype, AEL phenotype, and there are a list of genetic variations which are associated with this particular phenotype. So we have a list of genetic variations and their associated phenotypes which are systematically provided by the ISBT resource. This illustration or this tabulation will give a very clear picture of this particular concept. So if we take what is the gene, we have the ABO gene, and in this particular gene, there are a total of 149 variants, all types of variants. And all of these genetic variants are responsible for the expression of two major antigens, the A and B antigen. And since all of this is happening in ABO gene, they are categorized into a blood group system called as the ABO blood group system. And in this blood group system, there are a total of 13 phenotypes, including the A1, the A weak, the B weak, cis AB, everything. And this is a very quick example of how a particular genetic variant is associated with a particular blood phenotype. And one important point to be noted here is that it is not just one genetic variant which is associated with one particular phenotype. There could be a list of multiple variations which can be associated with one particular blood phenotype also. So all of this information have been systematically maintained by this pioneering body called as the International Society of Blood Transfusion by a very dedicated working party called as the Red Cell Immunogenetics and Blood Group Terminology Working Party. Now coming to the end of this session, let us quickly have a summary of what all we learned in this session. So the first thing is that the expression of RBC antigens are genetically controlled and they follow a Mendelian pattern of inheritance. And as of today, we have 51 genes which are associated with the human blood group phenotypes. These are called as human blood group genes. And in all of these 51 genes, there are more than 1,800 genetic variations which have been reported and validated till date. And all of this are corresponding to more than 600 blood phenotypes in an individual. And together, all of these findings are together systematically categorized into a total of 44 human blood group systems. Having said that, in all of our upcoming courses, we will be learning about how these genetic findings are going to help in the improvement of blood group profiling, and how is genomics going to play a role in advancing the future of these blood group profiling techniques, and how are we going to extrapolate on a population scale, which would help in developing population-specific blood group registries. So stay tuned for the next lecture, and thank you.